Welcome to GEN615. We are at week 1, Introduction to Teratology and Human Development. Learning objectives this week are going to be accomplished by the 2-day seminar weekend that you are going to have on March 3rd and 4th, as well as the materials in this course. Also, I have included the PowerPoint for this week from our speaker on teratology on Saturday. It's not narrated, but it's there, and you can. I'm going to open up this presentation so that you can print it out before she comes. So if we look at the learning objectives, it's to list principles of teratogenesis, discuss environmental factors associated with teratology, to review the torch studies, to review meiosis, normal female and male reproductive anatomy, follicular development and ovulation, and to discuss factors that influence the viability of gametes. I'd like to begin by looking over the reproductive systems of the male and female. And if we start off by looking at the reproductive structures, the terminology is they're gonads, and gonads are organs that produce gametes as well as hormones. The reproductive systems of both the male and the female have ducts, and these ducts receive and transport the gametes. They also have accessory glands that secrete fluids into the ducts, and the perineal structures are collectively known as external genitalia. The term reproductive tract is a term that's used for all the chambers and passageways that connect the ducts to the exterior of the body. And if we look at the male and the female reproductive system, they're functionally different. A female produces 1 gamete per month on average and retains and nurtures the zygote, whereas the male produces large quantities of gametes and in fact produces about 500 million sperm per day. Looking at the male essential organ for the male reproductive system is the testes. They secrete male sex hormones, which are called androgens, and they also produce the male gametes, the spermatozoa or sperm. The spermatozoa have a specific pathway or route that they follow from the interior of the body to the exterior of the body. They are formed in the testes. They then go to the epididymis, then to the ductus deferens, which is also referred to as the vas deferens, then to the ejaculatory duct, and then to the urethra. This figure is showing the main structures of the male reproductive system. Starting from a testis, the spermatozoa travel within the epididymis, then they go to the ductus deferens, then they go to the ejaculatory duct, and then the urethra before leaving the body. However, there are accessory organs, the seminal glands, the prostate gland, the bulbourethra glands, and they secrete various fluids into the ejaculatory ducts and urethra. The external genitalia consist of the scrotum, which encloses the testes and the penis, and the distal portion of the urethra passes through the penis. The slide is showing you the accessory glands, the seminal gland, the prostate gland, and the bulbourethra glands. Sometimes you will also hear the bulbourethra glands referred to as the Cowper's glands. So when we look at meiosis, remember meiosis is a special form of cell division which produces the gametes. And when we look at meiosis, when we produce the sperm in the male, the spermatozoa, then it's referred to as spermatogenesis. When we produce the eggs, the oocytes in females, it is referred to as oogenesis. Remember that the gametes contain 23 chromosomes, half the normal amount, And also remember that during the process of spermatogenesis and oogenesis, 4 haploid cells, each with 23 chromosomes, are produced. And that the fusion of the male and the female gametes produces a zygote with 46 chromosomes. If we look at the male first, we see that in the seminiferous tubules, meiosis begins with the primary spermatocytes, and in the end, it produces the spermatids, which are undifferentiated male gametes. This is a slide showing you a spermatozoon, and each spermatozoon has 4 distinct regions: the head, the neck, the middle piece, and the tail. And it's the head that contains the nucleus with the densely packed chromosomes. And at the tip, you can see on here, is the acrosome. And the acrosome or acrosomal cap contains enzymes. And these enzymes are essential in order to have fertilization. We can also see a short neck attaches the head to the middle piece, and the neck contains both centrioles, and the microtubules of the centrioles are continuous with those of the middle piece and tail. There's mitochondria in the midpiece. They're producing ATP to provide the energy for the spermatozoon to swim, to move. The tail is the only flagellum in the human body. And unlike other less specialized cells, a mature spermatozoon lacks an endoplasmic reticulum, a Golgi apparatus, lysosomes, perioxysomes, inclusions, and many other organelles. And by not having these organelles, it reduces the cell size and mass. And basically, what is the sperm? It's essentially a mobile carrier for chromosomes, and the extra weight would slow it down. Because the spermatozoon lacks glycogen or other energy reserves, it must absorb nutrients, primarily fructose, from the surrounding fluid. The surrounding fluid is the seminal fluid. So looking at the seminal fluid, it's the fluid component of the semen. It is a mixture of glandular secretions with distinct ionic and nutrient composition. It contains the combined secretions of the seminal glands, which makes up about 60% of the fluid, the prostate gland about 30%. It also contains nurse cells, which assist the sperm, and also the epididymis provides about 5% of the fluid, as well as the bulbourethra glands, giving the last 5%. So if we look at the seminal fluid, we see there's a high concentrations of fructose. This is what the spermatozoa will use for their energy source. There's also prostaglandins or tissue hormones, and what these do is they stimulate smooth muscle contractions in both the male and the female. And then we also have fibrinogen present, and it forms a temporary clot in the vagina to keep the sperm and egg in place. And it is slightly alkaline. And it's alkaline because you need to neutralize the acids in the prostate gland as well as in the vagina. So let's look at the female reproductive system. It produces sex hormones and functional gametes. It protects and supports the developing embryo, and it nourishes the newborn infant. Remember that the essential organ for the reproductive system of the female are the ovaries, or an ovary, you don't have to have two. And we look at the other organs of the female reproductive system, the uterine tubes or the fallopian tubes, the uterus, the vagina, and the external genitalia. This is a diagram of a woman's reproductive system. And remember that a woman's reproductive system produces the hormones and the functional gametes. And it must also be able to protect and support the developing embryo. And in this picture, you can see the ovaries, the uterine tubes or fallopian tubes, the uterus, and the vagina, and the components of the external genitalia. As in males, a variety of accessory glands also release secretions into the female reproductive tract. This is another view of the female reproductive tract. One of the things I'd like to point out is if you look here at the uterus is a very, very strong muscle, and it consists of the three layers, the perimetrium, the myometrium, and the endometrium. Looking at the ovaries, they're small, lumpy, almond-shaped organs, and they're near the lateral walls of the pelvic cavity. They perform three main functions: produce immature female gametes or oocytes, they secrete female sex hormones, and they secrete inhibin, which is involved in the feedback control of the pituitary follicular stimulating hormone production. Each ovary is stabilized by a meso-ovarian and by a pair of supporting ligaments, the ovarian ligament and the suspensory ligament. And you can see in here the posterior view of the uterus, the uterine tubes, and the ovaries. And this is the posterior view of the uterus, the uterine tubes, and the ovaries. And then you can see pointed out here the ovarian ligament, the mesovarian, and the suspensory ligament. If we look at this sectional view of an ovary and the uterine tube and associated mesenteries, you can see the nodular consistency of the ovary and the visceral peritoneum, which is also referred to as the germinal epithelium, covers the surface of each ovary and consists of a layer of epithelial cells that are columnar in shape that overlie a dense connective tissue layer called the tunica albuginea. And then if we look, we can divide the interior tissues of the ovary into a superficial cortex and a deeper medulla. The gametes themselves are produced in the cortex. If we look at this slide labeled number 1, you can see the primordial follicles in the egg nest, and the primary oocytes are located in the outer portion of the ovarian cortex near the tunica albuginea in clusters called egg nests. A single squamous epithelial layer of follicle cells surrounds each primary oocyte within an egg nest. The primary oocyte and its follicular cells form a primordial follicle. Beginning at puberty, primordial follicles are continuously activated to join other follicles already in development. The activating mechanism is unknown, but local hormones or growth factors within the ovary may be involved. The activated primordial follicle will either mature and be released as a secondary oocyte or degenerate, and the process where it degenerates is called atresia. This monthly process is known as the ovarian cycle. If we now take a look at the formation of primary follicles, we see that the preliminary steps in follicle development vary in length, but they can take almost a year to complete. Follicle development begins with the activation of the primordial follicles into primary follicles. The follicular cells enlarge, divide, and form several layers of cells around the growing primary oocyte. These follicular cells, which become rounded in appearance, are now called granulosa cells. Microvilli from the surrounding follicular cells intermingle with the microvilli from the surface of the oocyte. This glycoprotein-rich region is called the zona pellucida. The microvilli increase the surface area available for transferring materials from the follicular cells into the growing oocyte. As the granulosa cells enlarge and multiply, adjacent cells in the ovarian stoma form a layer of theca cells around the follicle. Theca cells and granulosa cells work together to produce sex hormones called estrogens. If we look at number 3, it's the formation of the secondary follicles. Many primordial follicles develop into primary follicles, but only a few of the primary follicles mature further. This process is apparently under the control of a growth factor produced by the oocyte. The transformation begins as the wall of the follicle thickens and the deeper follicular cells begin secreting small amounts of fluid. This fluid, which is follicular fluid, accumulates in small pockets that gradually expand and separate the inner and outer layers of the follicles. At this point in time, this is known as a secondary follicle. The primary oocyte continues to grow slowly while the follicle as a whole enlarges rapidly because the follicular fluid is accumulating. We go to the formation of the tertiary follicle. This is about 8 to 10 days after the start of the ovarian cycle. The ovaries generally contain only a single secondary follicle destined for further development. Now, by the 10th to the 14th day of the ovarian cycle, that follicle has become a tertiary follicle or mature Graafian follicle. This complex spans the entire width of the ovarian cortex and distorts the ovarian capsule. And what it does is it creates this bulge in the surface of the ovary. The oocyte projects into the antrum or expanded central chamber of the follicle. And this area is surrounded by a mass of cells called granulosa cells. Until this time, the primary oocyte has been suspended in prophase of meiosis I. As the development of the tertiary follicle ends, hormones levels rise of luteinizing hormone, prompting the primary oocyte to complete meiosis I. Instead of producing two secondary oocytes, the first meiotic division yields a secondary oocyte and a small non-functional polar body. The secondary oocyte then enters meiosis II, but stops once again at metaphase. Meiosis II will not be completed until fertilization occurs. Generally, on a day 14 of a 28-day cycle, the secondary oocyte and the attached granulosa cells lose their connections and drift free within the antrum. The granulosa cells still associated with the secondary oocyte form a protective layer known as a corona radiata. On this fifth slide, which designates ovulation, the tertiary follicle releases the secondary oocyte, and the distended follicular wall suddenly ruptures, ejecting the contents of the follicle, including the secondary oocyte and the corona radiata, into the pelvic cavity. The sticky follicular fluid keeps the corona radiata and the oocyte attached to the surface of the ovary. The oocyte is then moved into the uterine tube by contact with the fibrae of the fallopian tubes that extend from the tube's funnel-like opening, or by the fluid currents produced by the cilia that line the tube. Ovulation marks the end of the follicular phase of the ovarian cycle and the start of the new phase, which is called the luteal phase. In order to have fertilization to occur, the secondary oocyte must meet the spermatozoa during the first 12 to 24 hours. Fertilization typically occurs near the boundary between the ampulla and the isthmus of the fallopian tube or uterine tube. The uterine tube provides nutrient-rich environment by secretions from cells that are called PEG cells. And these cells contain lipids and glycogen. The nutrients supply the spermatozoa and developing pre-embryo. Now, in order to move that zygote down to the uterus, it involves ciliary movement and peristaltic contractions in the walls of the uterine tube. And in fact, a few hours before ovulation, actually, the hypogastric plexus nerves turn on a beating pattern. And what this does is it initiates peristalsis. It normally takes about 3 to 4 days in order for the zygote to reach the uterus. The uterus provides for the developing embryo, and it provides for the embryo, which is weeks 1 through 8, and for the fetus, which is week 9 through delivery. It gives it mechanical protection, nutritional support, as well as waste removal. Please go to the link to the self-study review embryology course from the universities of Freiburg, Lucerne, and Bern, which was developed for medical students. Go to the embryogenesis portion and then go to chapter 3, gametogenesis, and then you just go through the review there for some of the things that we just talked about. Just like to review the torch studies. We talked about it before, but I just want to go over a few things with this since we're going to be having this presentation this week and talking about teratology, beginning to talk about teratology. TORCH is an acronym for a group of infectious diseases that can cause illness in pregnant women and may cause birth defects in their newborns. And the TORCH panel is a group of blood tests that detects the presence of antibodies produced by the immune system in response to these infections. And then confirmation of an active infection may require more specific tests. So looking at what the tests are that make up the TORCH panel, we have T is toxoplasmosis, which is a parasitic infection that can be passed from the mother to the baby through the placenta during pregnancy. It's an infection with Toxoplasma gondii, which is a protozoa. It can be acquired during pregnancy through cat litter, infected cats, through drinking unpasteurized milk, goat's milk primarily, or eating contaminated meat. The O stands for other, and it can be various things such as syphilis, hepatitis B, HIV, Epstein-Barr virus, varicella-zoster virus, and parvovirus, B19. And then if we look at the rubella is the virus that causes German measles, the 3-day measles, which if you contract this early in pregnancy, an infant may develop heart disease. The growth may be stunted, hearing loss, blood disorders, vision problems, or pneumonia, central nervous system problems. Then the cytomegalovirus CMV is another viral infection that a mother may have acquired. Actually, more than half of all American adults have been infected with CMV at some point in their lives. And in most cases, it doesn't cause a severe illness. However, it may be passed to the baby during the birth process and can also infect newborns through breast milk. And some of the problems associated with the CMV are hearing loss, vision problems, mental retardation, pneumonia, and seizures. Now, herpes simplex virus is common. The two most common infections are the herpes simplex cold sores and also genital herpes. It was HSV1 for above the waist, and for below the waist it was HSV2. However, there has been contamination, cross-contamination. Newborns who contract the virus usually do so during traveling through the birth canal of the mother who has a genital infection with HSV, and then it can spread throughout the newborn's body. It can cause death, and most of these surviving babies, even if they're treated, have permanent damage to their central nervous system. So I just wanted to go back over this because I will be referring to this, and also it's a discussion that will occur in your presentation this week on teratology. So the last thing I need you to do for this week is I need you to do that one assignment, which is a case study. And then we will continue on next week with going through the first 3 weeks of gestation.